So today I'm going to talk about developing a novel NTRK1 inhibitor using virtual screening and a kinase inhibition assay.
I have a question.
All right, what's up?
Always wondered, how do you pronounce your last name?
Kwitchoff. It's like switch off, but with a K.
That's what I thought. Like,
Yeah.
That's not right.
You'd be surprised how many people actually mess it up. It's actually kind of really upsetting.
Switch last name, but I get it.
Yeah. All right. Continuing on. So it worked. So for the background, NTRK1 acts as a kinase, which activates when nerve growth factor B binds and signals for autophosphorylation of the kinase. The activated kinase then goes on to phosphorylate other proteins and elicit signal cascades essential for the proliferation and survival of neuronal cells, specifically sensory neurons. Papillary thyroid carcinoma typically expresses through mutations in the NTRK1 gene to form TRK oncoproteins. These proteins do not require the presence of NGFB for activation, as they are perpetually active. This leads to unregulated cell division and the development of cancerous tumors. The pharmacotherapeutic entrectinib was an effective inhibitor for this particular TRK. However, mutations in the proteins such as G667C and G595R have conveyed drug resistance that renders entrectinib ineffective. This phenomenon is not exclusive to this type of cancer, and new cancer therapeutics must be discovered faster to compensate for the rate at which the disease expresses mutations. So, the overall objective is to discover novel compounds used to inhibit enzymes essential to the life cycles of different cancers through high-throughput virtual screening methods. And just as an example, I have images from high-throughput docking of the different mutants with different drug ligands. So for the methodology, I have my workflow on the side here, where we're going to go through the Zinc 15 library and the FDA drug libraries, which is like millions upon millions of different compounds. We're going to go through GLIDE high throughput virtual screening and take the top 10% of those that pass through the screening. And then we're going to move on to GLIDE standard precision docking, take the top 10%. And I noticed that there's a typo that says top 100% of the GLIDE extra precision. That's not true. We're taking the top 10% of those. And then based on those, we're going to run a protein kinase or a kinase inhibition assay. And each of these scores are based on the relative docking scores, star number, and human oral absorption percents. I don't have those numbers because the bioinformatics student paper that I was assigned, that got messed up. I don't know how. And none of the figures were actually readable, so I couldn't get any of the data from it. So for the novelty and innovative aspects, the primary novelty and innovation is computational modeling versus trial and error, so the new way versus the old way. And it is faster and less expensive than traditional drug discovery process, which, as Jesse was talking about, takes between 12 to 15 years and a couple $1 billion, which is just completely ludicrous, where you can shorten that time immensely by using high throughput screening methods. And just as an example of how fast it can be done, we already found the top 10 compounds. The top 5 are from the FDA compound library, and the bottom are from the Zinc 15 compound library. And then the research plan and expected results is now that we have those compounds, what we're planning on doing is a kinase inhibition assay. So the goal is to find the IC50 of drugs against the mutant variant of kinase. We'll compare the known IC50 of the FDA-approved drug to the experimental IC50s of the new compounds. And then once we do that, the actual assay itself is comparing 5 different concentrations for the 10 different compounds, where we have a blank, positive control, the 10 times reduced IC50, just the normal IC50 from the literature values, and then the 10 times greater IC50. And then overall, we want a lower IC50 for the mutant type and a higher IC50 for the wild type, and that's because wild type proteins have natural functions, and if we target them, you could get off-target effects, like bad side effects that we wouldn't want. So yeah, and these are my references. Questions, comments, concerns?
Tell us more about your last name.
It's Polish. I don't know what. Yeah, that's about it.
You like the boss?
I do. It's good. Yeah. All right. There you go. Yeah, I'm pretty Polish.
I'm Polish also.
Oh, there you go. The more the merrier. What's up? No what?
I used to get on the stuff, so I was curious.
So specifically, I did not find any other inhibitors. But yeah, that was the only inhibitor that I came across. Like my assigned drug was the only inhibitor that I came across. And specifically, this this particular mutation occurs in like thyroid cancers and like brain cancer, stuff like that. So because it's like nerve growth factor. So it's really for a lot of like nervous system based cancers. So yeah.
Thank you.
Okay, so for your title, I want your title to be including the wiring thing. Okay, so then, oh, yeah, so that's good. And the timeline. So you propose this.
Yes.
Then, and once you estimate the timeline, so really it's already finished the dry lab portion. So for the dry lab, if you estimate how long does it take and what the agent will need and to pass the ICP for the five compound plus the two protein plus the listing drug. So you are done.
You want me to give you a timeline estimate right now? I would say honestly anywhere between like 1 and 3 months. I was reading, so around about that. And in terms of the reagent, it's the reagents that we used essentially for the wet lab. I can include that in the paper, obviously, but okay.
Okay, good.
All right.
That's why I don't bring one to the max.
What's every? Korea's going.
Oh.
Oh my God. My sibling don't say anything special, but I don't know if it's anything good. Yeah, it's my